Good morning. I'm sorry to be giving my talk remotely. I'm actually here in Washington, but have a faculty meeting to select next year's fellows that I have to be present for, and I'll be joining you in person later in the day. Now I'm going to talk to you about measuring antiviral activity in antiretroviral drugs. The issue is really, what did we learn from our use of genotypic and phenotypic testing for traditional small molecule antiretrovirals, and how might that inform our discussion about the role of susceptibility testing in the use of broadly neutralizing antibodies for the treatment of HIV. These are my disclosures. So let's start with a discussion of genotypic testing for drug resistance. Genotyping, of course, determines the presence or absence of specific changes in HIV genes encoding the drug targets, such as capsid, protease, reverse transcriptase, integrase, or envelope. But because HIV is so variable, we can't simply take the presence of a nucleotide or amino acid substitution compared to some consensus sequence as evidence of resistance. The interpretation of a genotype presupposes knowledge of which are the critical mutations that actually confer resistance. And so we're really inferring drug resistance by the presence of these known mutations. Now, there are a variety of methods and platforms that can include Sanger sequencing or several of the next-gen sequencing approaches that may be used to generate this sequence information. But ultimately, these all require some kind of interpretive algorithm. Well, phenotyping, of course, measures directly the amount of drug necessary to inhibit viral replication in a tissue culture system. And this can be used to determine what the IC50 or the concentration needed to inhibit replication by 50%, or the IC90 by use typically of recombinant or pseudotype viruses. And these assays, especially when they are automated and done in highly replicatable manner, can be quite precise, measuring changes of just 1.5 to 4-fold. But the challenge here is interpreting the meaning of a shift in susceptibility. Clinically relevant breakpoints have not been determined for all of the antiretrovirals, and it's critical to remember that these assays merely measure drug susceptibility. The definition of drug resistance really requires clinical correlation. So there are many challenges in defining antiretroviral drug resistance. First of all, there are numerous reasons for the failure of ART. Not all of them have to do with resistance. There can be poor adherence, inadequate regimen potency. There may be pharmacologic factors that lead to inadequate exposure. And of course, there can be either transmitted or emergent drug resistance. There are then several steps that are needed to define antiretroviral drug resistance. Typically, this begins with the emergence of particular mutations in association with virologic failure in people being treated with the drug in early phase clinical trials, then demonstrating that the mutations observed actually are responsible for reducing drug susceptibility by introducing them through site-directed mutagenesis into recombinant viruses and reproducing the phenotype. It then becomes important to demonstrate that the presence of that mutation actually impairs treatment response, and to determine the fold change in susceptibility that's associated with reduced clinical efficacy. Now, this slide takes us back nearly 30 years to the very first study that demonstrated the clinical significance of a shift in susceptibility to zidovudine. This was a study done by Rich D'Aquila, the late Vicki Johnson, Tony Japour, Clyde Crumpacker, Doug Richmond, and myself, using samples collected from a study that compared continued zidovudine to a switch to didanosine in zidovudine-experienced people. This was ACDG 116B 117. And we had developed, with the help of Doug Mayer, a consensus phenotype assay that allowed us, with generally good reproducibility at the time, to determine the IC50 for zidovudine. And we could characterize viruses from the participants in this trial as either high level or moderate or susceptible. And what you can see is that the presence of high level resistance was associated with a significant increased risk of death in people who continued to receive zidovudine. Actually, this was independent of their treatment assignment, because even after controlling for assignment to didanosine, this risk persisted. Switching to didanosine reduced your risk, but there was still a relative increased risk compared to not having resistance. That's because we didn't understand there was cross resistance to didanosine as well. We could also show that presence of the combination of critical zidovudine resistance mutations, changes at codons 215 and 41, likewise had an increased risk of progression to the primary endpoint, as you can see here in the adjusted analysis, and a substantial increased risk of death, 3 to 5-fold. So that was evidence that resistance mattered, and what level of resistance or which particular mutations might matter. The situation is much more complicated when you start to look at envelope. And this is an example from ACTG 5111. This was a phase 2 study of an investigational CCR5 antagonist, Vicriviroc, a drug that never made it to full approval. But what's notable are several things. First of all, here we're looking at the susceptibility curves for 3 different CCR5 antagonists, TAK-779, Maraviroc, which is of course FDA approved, and Vicriviroc, for 3 different sequences obtained at the time of treatment failure from participants. And what you note is, instead of seeing a shift to the right of these curves, the curves are instead flattened, so that you fail to achieve 100% inhibition. And for Vicriviroc, you actually saw stimulation of replication. Sorting out which of these mutations is actually responsible for the phenotype is really very challenging and gets quite messy, as you can see in the panels on the right. Here, what we did was to take, for one of the participants, the week 28 sequence when the resistance was present, and back mutate substitutions at 2 key positions. And you can see here, looking at the Vicriviroc, that these filled diamonds represent the week 28 sequence, and the open squares show what happens when the 306P mutation is reverted to the wild-type serine. And there is some restoration of the curve, but you still only are able to achieve about 50% inhibition at the highest drug concentrations achievable in the assay. By comparison, a monoclonal antibody against CCR5 had very nicely behaved sigmoid inhibition curves. Now, the approach that's typically been taken to try to identify which mutations are important and what fold resistance is clinically significant has involved some very complex regression modeling, as was done for etravirine in the duet studies. And I show this slide only to indicate that these exercises are not for the faint of heart. But what the group from Tibotec did was to look at mutations that they had identified in phase 2 trials of etravirine, and then pool the data from the duet studies to look at correlations between the fold change in etravirine at study entry and the change in reduction of virus load over the course of the study. And you can see that there is a correlation, although it's a fairly rough correlation with a lot of points that fall way off of the regression line. And that can be for many reasons. So, for example, for those people who had susceptible viruses, but nevertheless failed to see much in the way of viral load reduction, perhaps that's due to non-adherence. For those who have high fold change, but nevertheless achieve a significant reduction in virus load, perhaps that's because of the activity of boosted darunavir in their regimen. So when you try to put this all together, you can do an analysis where you explore the proportion of participants who achieved complete viral suppression by the Tullover analysis, and how that related to the fold change in IC50 for etravirine. And you can see that you don't really get much better than 70 to 77%, whereas those who have the highest level of resistance are only at about 37%. You can also do the same analysis for the mean change in virus load by category, where there's a clearer stepwise relationship. And that's been used then to try to categorize the impact of the weighted etravirine genotype score based on the impact of individual mutations on the phenotype, and then categorizing them as those that were associated with the highest response. So you could have a score of up to 2.0. Those that had an intermediate response, where only half of participants achieved viral suppression, and you could have a score up to 3.5. And those who had a reduced response of just about 38%, which was really not much different in that trial from the placebo arm, the people who did not receive etravirine. But you can see that even within each group, there's a fair bit of heterogeneity in the response. Now, for those of you who are still looking at resistance test reports, and I use one here from Monogram just for purposes of illustration, you know that this can be quite challenging to look through. And here make the point that when we ask about cutoffs for susceptibility, those shown in bold are considered to be clinical cutoffs. That is, there are data showing that viruses that have an IC50 or a fold change above this bolded value have reduced or no response. When there are two numbers, it means the lower threshold and then the upper threshold. So in between, there might be expected some intermediate response. And the values that are shown unbolded are for drugs where we really don't have any biological cutoffs. The cutoffs are based on the so-called biological cutoff from the 95% distribution of susceptibility of supposedly wild-type viruses. But that makes it really quite challenging to be able to interpret the fold change in any given person's virus. So to conclude, identifying resistance-associated mutations and defining phenotypic cutoffs for antiretroviral susceptibility is quite complex. This complexity increases dramatically when considering hypervariable targets such as envelope. Interpretive algorithms require regular updating to incorporate new drugs and viral evolution, a major problem when phenotypic tests are not ordered routinely from the clinic. And updating algorithms requires a regular flow of samples for testing. So with that, I will conclude, and although I won't be part of the discussion immediately after in this session, I hope to be able to catch up with you later in the meeting.